It's where we are looking at this right here. So we got big idea. Let's polish it out here. So we're going to go step by step on what we're looking at here. So like I said, if we take a look here, this maps it out everything here. So the very first thing here is the unzipping. And so an enzyme called, like we talked about, the helicase unzips the double-stranded DNA. It's breaking that double bond, the hydrogen bond, okay, between the complementary base pairs, separating it into separate strands. So that's a key thing about the helicase, is that it's breaking hydrogen bonds. All right, in fact, that's so nice, I'm gonna highlight it. Boom. Okay. And so if we go ahead, we can go ahead and draw our helicase here, or label it anyways, helicase. All right. Now, here's the deal. It's not as important for us in class, but I'll mention it here. One of the problems about this DNA is that when it unwinds, because of the structure, it wants to go back into that spiral and it wants to fold in on itself and try to reconnect the separated strands, because adenine doesn't want to be by itself and thymine doesn't want to be itself, same with G and C. So when the DNA is doing its thing, it has these enzymes that work to try to stabilize it. And there's a couple of different ones I want to work on. The first one is what we call, and it's a $5 word, topoisomerase. Okay. And what we call single-stranded binding proteins. Whoops. Now, single-stranded binding proteins attach to the unzipped DNA to stop it from reannealing or joining back together. The enzyme topoisomerase, it helps relieve that tension by relaxing it to its supercoiled structure. Okay, essentially, it prevents the single-stranded binding protein prevents it from coming back together, and the topoisomerase kind of helps allow it to go back into that double helix trait. All right. So we have our stabilization. Now it's time to go ahead and start priming. So we're going to be starting on, let's first orientate where we are in this particular case. Okay, so if we're looking at the picture, try to figure out where the 5 primes is, where's the 3 primes, and all of that. And so let's kind of take a look at this right here. So I'm looking at this strand. Is this my strand, the leading strand, or the lagging strand? And so if I follow the 3 prime, all right, the 5 prime end right here is what's going to be exposed. So in this particular case, this is going to be my lagging strand. So let's kind of go ahead and let's label these, and who are we looking at in this particular case here? So the actual primers that are being made, that's the Okazaki fragments. Again, those are the primers that that allow the lagging strand to be able to be replicated here. Here we have the ligase, which we'll find out is the glue here. Here is the exonuclease. Not nucleus, but nuclease. And then over here we have the primase. And we have the RNA primer. And of course, we have our DNA polymerase. All right, so let's go ahead and let's kind of step through this here. So the priming, all right? So the primase is an enzyme that attaches the RNA primers to the DNA, providing a starting point for the polymerase. Okay, so you have to have that 3' end to be able to replicate. Now, take note, what direction is the replication occurring? Replication's occurring in this direction. So both are overall going in this direction, okay? All right, so the next one is going to be, so we got to go look at number 4, and that's going to be the building and the extending. So the DNA polymerase, the DNA polymerase associates the DNA and builds the new DNA strand by adding new DNA nucleotides. It can only build the strand into the 5' to 3' direction, creating a leading strand and a lagging strand. And so we can go ahead and label our leading strand here. All right. Now, we got that. Now it's time to start replacing all the RNA nucleotides. And so here's the deal. Here, once it starts. It's continuous for that leading strand. There's really nothing that needs to be done. The problem is the lagging strand that has most of the problems associated with it. So this is where once it's copied and we have those primers down, we can't have RNA in there, okay? It's not going to read correctly. It's definitely not going to make protein. So we have to replace that DNA. So primers have to be removed by what we call the exonuclease. I'm going to go ahead and put that here, exonuclease. And again, this is all enzymes because they are made up of RNA. And the DNA polymerase then replaces them with DNA nucleotides. So this exonuclease, wherever we put that RNA primer, it goes, gets rid of that primer, and then that DNA polymerase comes back in, adding those particular things. Now, those Okazaki fragments, those are the primers that we've been talking about here, okay? All right, now we got to make sure that all of our phosphodiester bonds are formed. I'm going to put this right here, phosphodiester bonds. So the enzyme, what we call ligase, joins or glues the DNA fragments together on the lagging strand, okay? All right, these are simply just DNA nucleotides. Now, wow, that was a lot, but so far we got the lead and lagging strand. Now, for both of them, and it's a good thing we do this, the last thing that actually happens is that it proofreads. It checks for any errors that may have occurred during any of the leading or lagging strand replication. So the DNA polymerase also scans the DNA and checks for mistakes with the DNA. It checks for any errors, and like a boss, it goes and fixes them as well. All right, so hopefully this helps a little bit on your understanding of DNA replication. If you have any more questions, please let me know. All right, welcome. In this lecture today, what we are going to be looking at or trying to understand is this, the how and the why DNA replicates. All right. Now, in the last lecture, we talked about some of the reasons on why why does DNA replicate? And DNA replicates anytime there is any sort of cell division. Anytime a cell needs to divide, this can be from growth, repair, or regeneration. So the one that we want to really kind of focus on is the how today, not the why. And so we have to go back to our cell here. And so if we take a look at our cell, just like we've learned before, you kind of look at the anatomy. We got our cell membrane, Golgi, and all that good stuff. But for today's lecture, we're going to be in a eukaryotic cell, and we're going to be in the nucleus here because that is where the DNA is located. DNA is too large to be outside the nucleus, and just too many things that could possibly damage the DNA. So let's just kind of imagine the cell gets a signal to divide. And so we're going to be going to the nucleus. In fact, we're going to go to just a single part of the strand, the nucleus here. So we have our double helix and all that good stuff. And so the very first thing that's going to happen is, and this happens at multiple points in the DNA. So when we're talking about DNA replicating, it's starting here, 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 here. It is really happening at multiple points of everything. And so we're going to focus on this point right here. And so this is the point in which we start to see our DNA starting to open. All right, okay. So the point at which we're opening here, every point we open, we call that the replication fork. And if you kind of think about it, you want multiple replication forks happening all at the same time, all over the place of the DNA, so that it can occur with much more efficiency than it would be just starting at one end and copying it all the way through. It just takes too much time. So we're going to focus on this part right here. Now we got to remember that these are exposed bases, okay? Okay. So the first thing that's going to happen is the DNA is going to unwind, all right? And that is going to be done by what we call our DNA helicase, all right? I'm not going to go out and write all of this stuff, okay? Because in the next page, we're going to go over the definitions and write all that good stuff down. I just kind of want to walk through this. So we have DNA helicases acting on both ends, unwinding the DNA, going in opposite directions, okay? Now, the next thing that usually happens is DNA, as we know, is in that double helix, so you have stabilizing proteins that kind of help keep it from re-annealing, from joining back together. You don't necessarily need to know that for this particular case here. But the problem is that we do not have anything that we need to start off with. And so we have our next player in here, which is going to be our DNA primase. Now, I'm going to make any kind of new sequence of here. We'll make it green, okay? Okay, and so what this primase is going to do, it's going to start a primer on both ends of the DNA bubble, okay? And what this does is going to add RNA primers to this. So the RNA is going to be different. It's going to have a ribose to it. And so the enzymes are going to know that this is an RNA nucleotide, not a DNA nucleotide, okay? Now, when looking at this, let's go ahead and let's label this. So this is where we need the 5' and 3'. And remember, if we kind of come back over here, if we kind of take a look, I'll come over here. Remember that when we had our molecule, our DNA molecule, okay? All right, so we have our phosphate, we have our sugar, we have our nitrogen base. Remember, we have this H, and then we have this OH. So remember, this is our 3' end, this is our 5' end. And so what ends up happening is, okay, DNA is always built in the 5' to 3' direction, meaning that it only adds to the 3' OH end. And so when we have the next phosphate group coming in, what we actually end up seeing as a result of it, whoops, too many oxygens. If you imagine this is my next nucleotide, what ends up happening is that hydrogen and that OH end up combining and leaving as water. And so what ends up happening is, use a red marker, but that OH and that carbon end up creating that phosphodiester bond between that oxygen on that phosphate and that 3' sugar. And it always goes, again, 5' to 3' in that direction. So let's go back to our nucleus here, okay? So in this case here, let's say that this end right here is 5', this end right here is 3'. And it's going to be opposite here. This is going to be 3', and this is going to be 5'. Remember, DNA works anti-parallel, okay? Now, when we're looking at this, I'm only focus on this half of the bubble, okay? Just to kind of make things a little bit clearer for you, okay? So we're just going to do one half at a time to make this sense, all right? So first things we got to do is look and ask ourselves, which is the leading strand, which is the lagging strand, all right? So remember the differences between the two. Leading is where it's going to be continuous. I'll go ahead and put that right here. So leading strand allows for continuous replication. And that's the reason is because that 3' OH end is exposed. Okay, that is the big thing. Now the lagging strand, however, okay, what it's going to have to do that we'll find out is going to use Okazaki fragments. Oops, I spelled it. To create primers so that it can do that 3' direction. So let's take a look here. All right. So first of all, we can see here is our 3' end. That's where my OH is going to be located at. And here's the deal. All right. This is going to involve our DNA polymerase. All right. I'll draw her right here. And so in this case, this is going to be a continuous replication this way. And so this is going to be my leading strand. Now here's the deal. All right, most of your diagrams that you look at and everything only picture half of the replication bubble, as you'll see in the next page. It's oftentimes how it shows up in a lot of tests and assessments and everything else like that. You got to know that this is happening in both directions. So when we're looking at this, this is a bubble. So here's my other 3' end. At this replication fork mark, this side right here, okay, is also going to be replicating in a continuous fashion because it's going to keep adding to that 3' end as a result of it. And so on the other side of this, this is going to be my leading strand because it's going to be adding that direction and continuous at the 3' end. Okay, now let's go ahead and take a look at the other side. Here's the problem. We have this 5' end and we have this 5' end, okay? We cannot add to the 5' end. And so this strand right here, all right, is unfortunately going to be our lagging strand. And I'm just going to represent it like this because as we're going to find out, we're going to have to make little primers to replicate backwards, so to speak, to be able to replicate the DNA. And so same thing over here, as it replicates towards this DNA helicase, it's going to be fragmented, and that's going to be my lagging. All right. So hopefully this is making a little bit more sense. But we at least can see what ends up happening here. Now, before we go into talking about the nitty-gritties of the different ones and everything, let's talk about why we have these Okazaki fragments, okay? So in just a moment, we'll map out this much more clearly in the next segment here, but you are going to have a enzyme called a RNA primase that's going to make these what we call Okazaki fragments, these little segments of RNA that is made ahead of the replication fork. Now, here's the deal. It does this because what it does is that it creates a free 3' end on that lagging strand. We'll do the same thing over here on both lagging strands. And so what ends up happening is that you have the RNA polymerase, and what it's going to do is going to see, oh, I have a 3' end right here. So what am I going to do? I'm going to attach, this is my DNA polymerase. I'm going to attach right here to my 3' end, and I'm going to start copying in this direction because that is my 3' end until it connects to that primer again, okay? And so if I drew another RNA polymerase, it's going to add a base here, going in that direction. And so it kind of does this backwards on both sides as it goes in the opposite directions. Now, in the leading strand, that's not an issue because that 3' end is already there, and it's already exposed, and so this is not going to be stagnated, but continuous replication as a result of it, okay? So hopefully that makes a little bit of sense on what is considered to be the leading and lagging strand of the DNA. This is happening in multiple points of the cell, okay? Now, when we come back, we'll talk about, how do we go from that DNA ultimately to protein and trait, but I want you to turn to your 4, okay, when we were looking at this, all right, a lot of times we only see it where it's only half of the replication fork, all right? So